Thank you, thank you. So, as you see, I'm already here to save on time because we are running late of time. So, two requests. Please, each speaker, no longer than 20 minutes. If you speak less than that, you get a bonus. You said 25. And 25, including discussion. And I know it's a birthday celebration, so you're welcome to make a birthday wish. No more than 1 or 2 minutes because we will have plenty of time to do this over lunch. First speaker, Professor Robert C. Gallo.
You're taking up my time.
Come on.
Yeah, you have to be assertive here. Thank you. So how do we do the slides? This one? Up and down? Okay, if I want one, I got a pointer. This is up and down, right? I need you nearby, shall I? Ethan, thank you for that wonderful introduction, very long and thorough. Well, I don't know if I should start or not start. Oh, okay. Well, first let me say I'm honored to be part of honoring friend Isaac, and always glad to be in Israel. At many stages of my career, I owe much gratitude to the people in this country. I'm going to be talking about something that's off base in this meeting, which is about cancer. HIV is not a cancer virus, and I'm going to be speaking about HIV. But indirectly, well, it is tumorigenic, but it's not a transforming tumor virus. And how it's tumorigenic may be by, yes, immune suppression, but there's also evidence that it is involved sometimes with one or more proteins in activating genes or in the entire viral expression of viruses that really are tumor viruses. But that's still not going to be what I'm focusing on. I thought that in the time limited, I could give a more circumspect talk focused on one particular topic, why we don't have a vaccine against HIV right for the moment, and the difficulties we face in the future. Before doing so, though, I wanted to tell you, Isaac mentioned our Institute of Human Virology in Baltimore, and every year we have a Lifetime Achievement Award since the Institute began virtually. The first one to receive that is here in the audience is Professor George Klein. But a few years back, Isaac Witz was given this award. It's given by the senior staff of the Institute with suggestions of people's names, and then debates and debates for senior staff meetings once a week over a period of time until there's a virtual, if not complete, unanimous vote. So as I said a few years ago, Isaac received this award, and we had a picture that I told Isaac should be for posterity. And it was this picture. And Isaac said, this is a great idea. I'd like to keep this for posterity. But a little bit later, he came back to me and said, it could be improved for posterity. And he did that. But then another week went by, and he said, you know, I have further ideas about this. He said, for posterity, it really should be this. And we agree with him, so that's the picture that'll be in our institute for his award, and the picture for posterity from that day. Now let me jump into the story that I want to go over with you rapidly, because Itan now gets serious. We've got to be on time, we've got to be on time. It begins now, folks, so following speakers, be prepared. He's going to be strong. So a vaccine against HIV can't be based on principles, most of the principles of old, or let's say previous vaccinology principles. I first recognized this in 1985. I went to see Jonas Salk because there was nobody responsible, just like there was nobody really responsible for finding the cause of AIDS, and people got into it by whims or chance. And I asked him his view, and he told me ever since you learned how to grow the virus in permanent culture in '83 that you have a vaccine because you just have to fix it in formalin as they did for the polio vaccine. And I knew then we were really alone because the people doing classical vaccinology then and now were not familiar with the problems of a retrovirus. This means it differs in very critical ways from viruses we have developed successful vaccines for. The critical differences are it's got a very error-prone DNA polymerase, the reverse transcriptase, generating a lot of variability, and this is not the only way it undergoes variability. More importantly for me, and I've said this from the early years, is the fact that it integrates, and it does so within one day. Within 24 hours, the genes are integrated forever, and that has serious consequences for a vaccine. It's soon establishing infection and providing very little time for immune recall. Think of polio again. If we're all vaccinated for it, but we drink a vial of polio, we get infected for the most part. But your immune system says, hey, I've seen this before, I was vaccinated, and you have time for an immune recall, and you clear the enterovirus from where it's in, namely the gastrointestinal tract, before it gets to the brain and spinal cord. With HIV, it's too late. As I said, within a day, it's integrated, and within a few days, it's generating virus, and within a week, it's already generating variants of the virus. So I could already think, if you're with me, that a vaccine based on cell-mediated immunity is not going to work, even though that was one of the first major clinical trials. It shouldn't work. It's too late. You're waiting for cells to get infected. Variants are already being generated. We really need antibody, and we need sterilizing immunity. And some of these things have never been required before in the history of vaccinology. So infection is established quickly. That's the consequence. Permanently, no time for immune recall. A protective immune response, therefore, must persist. That's not been required before. And a complete block of infection, namely sterilizing immunity, in my mind, is a requirement. Never before a requirement for a successful vaccine. So the third and another difficulty is that we need a subunit vaccine. The FDA will never accept an attenuated virus nor a killed whole virus. And in any case, a killed whole virus modifies the surface protein, the envelope protein, the GP120, which is the first thing. If my hand is the virus and these are the envelope molecules, the first thing the cell sees is the envelope. That's what we have to target, GP120. But if you kill the virus, that's a modified protein, and it doesn't induce the right kind of antibodies. So we have to use a subunit. But if you look at the last part, subunit vaccines have been developed. Think of the late Morris Hilleman of Merck and the development, for example, of hepatitis B vaccine based on just a subunit approach. So the question is, was Albert correct? I used this slide because about a week and a half ago there was a celebration of Jonas Salk's 100th birthday, and it was also a celebration for Albert Sabin at the same time. So this is a talk, in fact, I prepared for that meeting with slight modification. So before Albert Sabin died, he wrote a letter to Science saying that a vaccine was impossible for HIV. So if he was right, then the people working on it, that would certainly include me, because I spend part of my time doing that, would be irrational, which is very possible. If not. Now, what have been the vaccine trials to date? There have been 6 major trials, at least from the United States. These have been the big trials. And they involve 4 different approaches, so 2 overlap with 2 on there. One was based from NIH solely on cell-mediated immunity, and as thought, this was completely ineffective. Another was, again from NIH with Merck, and it was adenovirus-delivered HIV immunogens with or without antibody generation, and it actually increased infection. Not a well-known story. It didn't get a lot of publicity, but the vaccinees got more infected. You may say, well, why? Was antibody generated that facilitated infection? I think not. I think what happened is that the adenovirus vector caused the problem by activating too many T cells, that the person had already seen adenovirus before. They got a lot of T cell activation. And where does HIV live? Inactivated T cells. That's a key problem that I'll end my talk with to tell you what our dilemmas are. Another trial, actually it was the first trial from a company called VaxGen, was kind of rational. Let's use GP120 itself for the vaccine. But this trial failed completely also. Didn't increase infection, but failed. And I think the reason for that was maybe obvious also. They use conventional GP120, which is hyper-variable. So if I use virus, let's say poor Josh Fiedler was infected and I used a virus from him. Anyway, his GP120, he'd be protected. And maybe a few of his friends that had the similar virus. But that's it. And I think that's what happened in this case. You get type-specific immunity. Now, the last is the trial by the United States Army with TIE. And they had the first and only partial success. And by chance, they used a modified GP120, modified in a way that we think was important for the success of that vaccine. Modest though the success was, and I come back to that in a second. The approaches from our institute are as follows. The vaccine should induce antibodies. Those antibodies should be protective. To be protective, they should target the glycoprotein 120,000, GP120, the envelope protein that sticks out there, because it is the first HIV component which the cell sees and which initiates infection. The antibody has to be broadly reactive, targeting conserved regions of the envelope protein, functionally required for infection. Now, I told you that the envelope protein is hyper-variable. The virus is variable. This guy is really variable, but yet there has to be some conserved regions. I mean, you can have, a lion can't be a giraffe. Something has to be common to the envelope protein. Those conserved regions that are required for infection, however, are very hidden. They're hidden by two ways, three ways. First, this is mobile, very mobile, so the antibodies have a hard time finding sites. Secondly, it's covered by a glycan shield. Thirdly, the envelope is not like that, but think of a ball of string curled in many ways, and the inner domains of this protein are hidden, and there lies the conserved important regions that the virus uses for infection or to first stage of infection. So the sites are hidden by protein folds, a glycan shield, and the mobility of GP120, the summary in the bottom of the slide in orange. Okay. So the approach we used was opening these internal hidden sites, if you will, by methods which also fixed the mobility of the molecule, allowing antibodies to bind several new epitopes. Well, how was this done? Well, first let's take a look at HIV entry. As you see on this slide, the virus with the lollipop on its surface, the lollipop is the GP120 molecule, at least the round ball is, and you see in the bottom CD4, and you see the co-receptor for HIV, CCR5, a chemokine normal receptor molecule. So the envelope protein first interacts with CD4, and then it undergoes major conformational change, which the late Don Wiley called the most significant change when one protein meets another in known structural biology. When that occurs, it initiates the fusion event shown over there, where the guts of the virus are entered into the cytoplasm of the cell. Let's see this by another cartoon from Robert Doms in Pennsylvania. So there's HIV, there's the GP120, the ball, interacting with CD4. I show it now as an aura because of this great structural change. So the structural change is shown from an oval going to this bean. That allows it to find CCR5 to initiate the fusion events for cell entry. What's our vaccine approach is in fact the bean. The GP120 in this shape bound to the domain 1 and domain 2 of CD4. Actually, there's a linker connecting them so the proper folding can occur, allowing the GP120 to properly bind CD4. So there is the vaccine candidate, which we'll begin clinical trials with in about 4 months' time, sponsored by the Gates Foundation. But I'll come to you what the problem is, even though we have some nice primate monkey, that is, data. We use this GP120. Here's the linker of 20 neutral amino acids. There's D1, D2 of CD4. We do it again in monkeys because we want a CD4 of monkeys as well as human, because if we're going to do monkey experiments, we don't want to have the antibody attacking CD4 and confusing our results. This is a summary of 7 different primate experiments. One of them, we didn't get any protection at all, and I'll come back to that in the end. So if we use a single challenge of a high dose, we get something like this. And you say, well, this is nice results. You can see it splashed in Nature, some publications, like Picker's results that have a lot of attention to them. The vaccine group has protection, but these animals are still infected. And my belief is that this is worthless, that this guy is going to come back eventually and it is of no consequence. So this is a single rectal high-dose challenge in a monkey with, the monkey virus SIV, or the hybrid called SHIV, that's HIV, which is a combination of HIV and SIV. But now we went to an experimental protocol that people say more imitates human infection. Multiple weekly challenges of low dose. So the greater the number of challenges, the better the candidate vaccine, still showing protection. So here's the control group. They all get infected. You get the peak and then it levels off, like over here in yellow. And the vaccine worked. We have sterilizing immunity. Bravo. We're very excited. Okay, but what happens? You keep challenging, we lose it all. This is exactly what happened in the human RV144 trial in Thailand. In the first months, they had a 62% protection That would be a licensed vaccine today if it lasted. But when you look at the results in a penetrating way, what happened is they completely lost protection, and it correlates with the fall of certain types of antibodies. Now you're going to say to me, those antibodies must be broad neutralizing antibodies. They're not. When you read, if there's any aficionado of the field here, be wary of the numerous papers in Nature, Science, Cell, in PNAS, and high-quality journals when they use homologous challenges. The virus you're challenging with should be very different. It's not always the case. It's meaningless to use a homologous challenge in my regards. I don't even look at the paper if it's a very similar virus challenge. That's one thing. And look if the results are like this rather than that. It also becomes relatively meaningless, at least the way we look at it. So that's a problem we're looking at. So the results of monkeys showing protection with the SHIV or SIV infection are reminiscent of the results of the one modestly successful HIV clinical trial known as RV144, the Army-tied investigators, in that complete protection was obtained, but it's lost in a short period of time as the titer of protective antibodies rapidly declined. Those antibodies with heterologous challenges in the entire literature, to the best of my knowledge, have never correlated with broad neutralizing antibodies. Indeed, it's almost impossible to generate antibodies that broad and neutralizing at any significant titer. And even when they occurred, they were not correlated with protection. What has correlated is ADCC. And I believe ADCC must be working not just only after the cell gets infected, but while the virus is attaching. And that's a lot of basic research in our institute right now about that very phenomenon. So this is the RV144 trial. Look at the top when it's plotted in months. Here's protection in black. And you can see terrific over here, right? It looks like a home run, almost, 60%, a licensable vaccine. But then beans, nothing, declines, with certain types of antibodies declining. Some of those antibodies have ADCC function. Now let's look at years. This would be the case of most effective vaccines today, which can go out for decades, even hundreds of years. This is HPV, human papillomavirus vaccine, does very nicely and continually. And look at what happens with the RV144. It just crashes. Isaac, do you agree with that? Or you're getting your rest now? That's okay. So how do we attempt to solve the problem of failure of these antibodies to persist? I mean, what would you do if you were us or you're in the field? And there may be somebody here in the field in more than one, and I know there's at least one. People try, or we have tried, various adjuvants, maybe vectors, maybe adding other HIV proteins. All of these approaches also are a double-edged sword. We need activated T cells for help to make antibodies, no question. But if we overshoot, we lose all efficacy. That's the lesson. Why is that? Our experiments are summarized on this slide. There needs to be an immune balance. We need to have some T cells, as shown in the bottom. This would be the mucosal surface, whether the vagina, urethra, anal tract. But if we have this, we're doomed. You get infection no matter what the heck you're doing. In fact, you wouldn't even know. You may have a very effective vaccine and wouldn't even know it because you activated too many T cells and you weren't able to see it. Don't ask me how you make such a plot. I didn't do this slide. This comes from my colleague George Lewis. I don't know what he even calls it, a hot thing or whatever. But all I know is this is important right here. You have to be in this region. So what are we looking at? There's 3 different criteria. The redder you are, the more protected. See over here? These are the number of challenges where we have protection. So you're out to 13 challenges, you're still protected. So what does it correlate with? The log of the titer of ADCC antibodies. The more you have, the better off. But look at the yellow spots looking at activated T cells. If you don't have any, you're doomed. But if you have too many, you're doomed. So there's a sweet spot. We call it an immune balance. That's the take-home lesson. This concept, these ideas are published in a PNAS perspective about a week ago, maybe 2 weeks ago. Antibody persistence and T-cell balance, 2 key factors confronting an HIV vaccine. I believe this is a problem for any candidate vaccine that has a chance of working. Even if you have the perfect immunogen, these are problems that need to be solved. They need basic immunology to help solve them. The conclusion for an effective HIV vaccine, even if we have the perfect envelope immunogen, as I've already said, lessons from primate studies and from the RV144 clinical trial, two critical basic science problems remain that must be solved: durability of the antibodies and a correct immune balance. I acknowledge my co-workers George Lewis and Tony DeVico of IHV for partnering in the concepts and in the work, and Tim Fouts of Profectus Biosciences, which was the spin-off company from our institute, and John Gershoni of Tel Aviv University, who contributed importantly to the early ideas, concepts, generating this approach to a vaccine. We owe NIH and especially the Gates Foundation for their scientific support. Isaac, once again, I shouldn't say once again because I haven't said it yet, a very happy birthday. Congratulations on your Hirsch lecture. Thank you for 40-odd years of increasingly close and much valued friendship. I learned much from you both in science and life in general. Please continue to be my guide and friend for years to come. And Nitan, thank you for having me with you.
Okay, we do have time for a short discussion. Are there any questions?
Can I say
You
something?
don't want to have
I start by an insult. I've never been in Israel before where I haven't had I like to say the lemon squeezed a little. So usually there's somebody jumping up and attacking.
Let's keep coming. You'll see more changes, okay?